Yeah, I have started. Welcome,
Okay.
Janis, for introduction to Bayesian metabolic control analysis.
Hi everyone. So I'm Janis. I'm gonna talk about Bayesian metabolic control analysis today. I just think that it's a cool method to use, and trying to extend it in my own research. So because this is an informational lecture, we do have some learning objectives. So basically, we're going to talk about the goal of metabolic control analysis, the particular use cases of Bayesian metabolic control analysis, and the general methodology of BMCA. So first of all, in the wider world, why would we even care about MCA or BMCA? And so the demand for sustainably sourced raw materials is increasing, and bio-based products are frequently produced through metabolically engineered microorganisms, but we really need to develop these engineering methods to be better and faster. So here's our current world. We're very reliant on petrochemically derived commodity chemicals, and we want to manufacture these commodity chemicals through biomanufacturing with more sustainable alternatives to petrochemicals. So what's holding us back is that these projects are not consistently commercially successful. They just take a long time and a lot of resources to make commercially viable. And so BMCA is one method in reducing strain engineering time and decreasing that resource load to make these more commercially viable. So if BMCA is successful, like if we were to run this method, what would we get out of it? So basically we would get a kinetic model of the metabolic pathway that we're interested in. And that would accurately predict which enzymes or reactions in the pathway have the most control. And if we know which parts of the pathway have the most control, we can develop more effective strategies on getting what we want from that metabolism. So here I wrote optimizing expression that can fall under increasing yield and titer, but it can also be things like decreasing side products or toxic metabolites, or using new feedstocks, more sustainable feedstocks, or stabilizing metabolism. So BMCA can be used for a wide variety of those applications. So the point of getting a kinetic model and the point of getting a kinetic model is to be able to predict which control points are, or where the control points are. And so this is just an example of how that's the case. Here we have a very small, let me try to get, okay. Here we have a very small pathway, and this is just an example linear pathway. And if I told you that these reactions E1, E2, and E3 follow the mass action rate laws, where K1 is the rate constants, K2 and K3, and X0 is the substrate concentration, and so on and so forth. And if I gave you the starting concentration, you would know like where these concentrations start. But without knowing these rate constants, you wouldn't exactly know how the species would play out over time. And so one way to address this challenge is to use metabolic control analysis to understand the metabolic pathway and know how they change within time. So metabolic control analysis provides a mathematical framework to describe metabolic pathways, and it quantifies the influence of network parameters on flux and species concentration through network-dependent properties such as control coefficients and elasticities. So using MCA, we can build kinetic models that can simulate how metabolite concentrations change in the system over time and further our understanding of the system. And MCA is very similar to BMCA. And so I just explained MCA, and for MCA, we need to, for it to work, we have to know like what the chemical reactions are, the whole pathway. And we also need to know we need to have the measurements that would help us predict the rate law. And so I put it very generally because it depends on which rate law you use. But most of the time, like we can use steady state perturbation data to applied to MCA. And so what steady state perturbation data is things that are, it's data that we collect. We allow the system to reach a steady state, and then we perturb it somewhere once, and then it reaches a second steady state, and then we collect like this change in steady state. So here we have, this is the pathway flux, and this is for E2 for this reaction. We start at time 0, we start at some initial state, and then it reaches a steady state, which I call J star. And then at this perturbation, when we change the amount or concentration of the enzyme E2, then the flux will changes some amount, delta J. And so from these steady state perturbation data, we can collect flux control coefficients. And that, like mathematically speaking, that's just the change in J over the reference J divided by the change in E2 over the reference E2. Or if you want to put it a different way, it's just a ratio of the change in J percent over the rate of change in E2 percent. And one thing to note is that data collected within the systems level context, data is collected within the system level context, and it doesn't involve isolating a protein in this kind of setup. And that's really crucial because there have been studies that show that data that has been collected at the systems level context when put into kinetic models it helps the kinetic models be more predictive than when we use data that has been collected from isolated proteins. So going back to comparing MCA and BMCA, we see that for BMCA, we also need a chemical reaction framework. And for BMCA, the measurements that we use are measurements of metallic concentrations, proteins, and reaction fluxes. And this is because, I'll show you later, but we use the LinLog rate approximation. And the input data, we also use steady state perturbation data. So BMCA is just an extension of MCA, and it applies modern probabilistic programming techniques to provide a way to fit more parameters at a genome scale. So here's an outline of the whole methodology. And so at the beginning, you can see that we're gonna choose a rate law, the LinLog rate law, for which we have to collect values for. But I kind of want to start at the very end of this as I go through this. So our goal for BMCA or MCA in general is to find the control coefficients. And so here's the flux control coefficients that I pointed out earlier. But in order to get the flux control coefficients, we need to get the concentration control coefficients. And to get the concentration control coefficients, we need to know what the elasticities are. And so the elasticities are something that we can find through Bayesian or BMCA. And elasticities are just basically how reaction rate changes when metabolite concentrations are perturbed. So going back to rate laws, there are many types of rate law approximations. I'm sure everyone's familiar with the Michaelis-Menten because it's used a lot in enzyme kinetics. And one thing to note is that in this Michaelis-Menten rate law, there are a bunch of parameters like this Vmax and Km that are really hard to measure, and they're harder to measure in vivo. However, we can, instead of using the Michaelis-Menten rate law, we can use a LinLog approximation. So this is a scalar form of a LinLog approximation. And it has lots of variables. So I'll go through them in the next slide. But the one takeaway I want from this slide is that here we have the Michaelis-Menten, kinetic approximation. And when you compare the LinLog, which is the dotted sky blue versus the power law versus the linear, we see that the LinLog actually follows the Michaelis-Menten pretty closely. But another takeaway is that it's very, the approximation works best near the steady state which is around here. So that's why we need to, when we get our measurements, it's best to get them at steady state, because that means that when we do the approximation, we're approximating here where the approximation is being made the closest. So back to the LinLog equation. Now I have this as the matrix form. And so the LinLog kinetics approximates a reaction rate V as a sum of logarithms. And these are all the logarithms of here, or the, I guess, just the terms. And one special thing about the LinLog as well is that it takes in, it can use relative amounts of concentrations as opposed to absolute concentrations. So here we have E divided by E star. So that would be whatever you choose your reference enzyme concentration to be, and these are just the rest of your enzyme concentrations. So then you get a relative enzyme concentration of E hat, and same for V hat. Here you have Y over Y star, and then we can log it so that it becomes a sum over here. So all in all, we have V, which is your flux rate, your E's, which are the rate of enzymes, or sorry, enzyme concentrations. These are values that correspond to the internal species. And here, this is the elasticity values that I pointed out earlier. So that's how, once we know our elasticities through BMCA, then that's how we can compute our control coefficients. And we also have the same for the external species. So these are the elasticities for the external species, and then the concentrations. And here's just a different breakdown. But this just tells you, like, in matrix form, what the matrix would just look like. The same thing. So given that rate law approximation, now that we know the variables that we're looking for, we want to collect the data. So what that means is that we would build a bunch of strains that all have the same genetic pathway, and then we'll alter the genetic pathway either through knockouts or through CRISPR activations or inhibitions in some way. And we'll just do one perturbation, just because it's easy to know and what the effect of that one perturbation is without thinking about interaction effects when you start doing multiple perturbations at a time. But we'll make multiple strains. So these are each separate strains that all have the same pathway. And then in each strain, there are different like knockouts or activations and stuff as shown here. So you'll make a library of different strains like that. And then you will, with your entire library of strains, you will start to measure like all the fluxes, steady state fluxes at a given time. So we're measuring this data by subjecting all of these strains to the same conditions. And we're measuring the fluxes, the enzyme levels, which is like proteomics, or we can measure the transcriptomics. They're not like one-to-one replaceable with each other, but if you can't get your proteomics, then you can use transcriptomics in a different way. We also measure the internal metabolic concentrations that we can, and the external metabolic concentrations. And one good thing about Bayesian inference or BMCA is that because it uses Bayesian inference, we don't really need to know every single data. Obviously, like having more data is better, because then we are able to fit, the algorithm is able to use more data points to fit things more accurately. But because it is Bayesian inference, it can compute uncertainties and kind of infer what those data points would be. So once you have the data, then it would just, our next thing would be to choose priors and fit the data for each variable into Bayesian inference. So what that looks like is we're just going to define a prior distribution here. And then given our data, we can define a likelihood function. And this likelihood is also, conditions on the data. And then from the two distributions, the likelihood and the prior, plus there is a normalization from the data, we get a posterior distribution. So that's this dark line here. Yeah. So each, I guess, variable in this LinLog approximation has its own, like we have to set up the priors in their own way. So for example, elasticities, their distributions are mostly half normals. So for reactants, the elasticity distributions are the half normal positives because reactants activate reaction rates, and vice versa for the products. And then regulators, which basically are, I guess, metabolites that are not directly participating in each chemical reaction. We're just going to guess that they're not participating, so their values are 0. For enzyme levels, we can use proteomic data, or if we have transcriptomic data, then we can also say that our prior is that we think that the distribution is centered at where that transcriptomic data value is, but we wouldn't really say that that's what we observe, like those are the values that we observed. Whereas for proteomic data, we would just say that's the value that we observed. If we haven't measured if we can't measure the enzyme data, then we just keep them at 0 or 1. We would use 0 if your data is like logged, because log of 1 is 0. But if you're not using a log, then you can use 1. And basically that means that that reaction is not different from the reference, right? So whatever value you have divided by the reference is 1, meaning that it's the same as the reference. And then we also have zeros for our enzyme priors, because those are just reactions that are outside of our system, and so we just kind of assume that they don't exist. And so basically there are 3 different types of enzyme priors that you have to take into account. And for metabolite and flux priors, if the values of the metabolites are not known, we just basically establish a distribution with a large variance. So it's going to just be squatty and wide. But if the values are known, we can use a linear solve to calculate this theoretical steady state values. And so that just involves like moving some of these what's called variables around. And that's just not shown here, but it's very simple math. And from that, I guess, the calculated steady state, we can use that as the prior and condition the prior on the data that we do have. So once we set up all of these priors, and then if we have the observers, then we get likelihoods. And once we get that, then the BMCA calculates these posteriors. And so from those posteriors, I calculate the highest density intervals. So it's not this, it's actually this thing. And I'm just saying this because it's nice to get like a range of what those values could be. But sometimes if you just want like a quick number, then it's nice to have like use the median, or just use the highest density intervals for those like 95% ranges. So just to clarify, the 95% credible interval is simply the central portion of this posterior distribution. And the confidence interval is something kind of different. It's just different. It's similar but different. So yeah, once you have all of your posteriors, so basically once you put in your BMCA for all of the variables, you get a bunch of like distributions for all of your variables. And then from those elasticities, you can calculate your metabolite or concentration control coefficients, and then your flux control coefficients. So we just went through this whole methodology for BMCA. And in summary, BMCA is an extension of MCA. Using BMCA, we can infer control coefficients from incomplete steady state perturbation data. And these control coefficients can be used to build predictive kinetic models, which can ultimately reduce strain development time and increase commercial viability of biomanufacturing projects. So yeah.
Thank you, Janis, for the clear talk about the BMCA. If you have finished, then maybe we can accept questions from the audience. Michael and Steve, thank you for coming. Do you have any questions?
Hello. How is it in practice? I mean, you mentioned the whole theory here, and it makes good sense, and it seems reasonable, but how is it in practice? Does it work out well?
I'm using it right now. I'm still haven't finished the benchmarking yet. I would say that in prior work, so like looking at what other people have done, qualitatively, it's pretty good. Like qualitatively, like it kind of is close to what measured values, but I can't really tell you yet exactly how good. So I think it's promising, but there are other things that we can do to improve. So if you've noticed, all of my distributions are Gaussians or normal distributions, and that is like an approximation we use, but maybe there are other ways or other approximations that are more, I guess, accurate, and that hasn't been explored yet.
Yeah, but those Gaussians, the distribution you get is whatever the posterior is. It could be any shape, ultimately. I mean, those Gaussians were just your graphics, just to illustrate what might happen.
Oh, so
the actual shape of the distribution won't be Gaussian. Not necessarily, anyway.
I thought that, okay, correct me if I'm wrong, but I thought that for ADVI, they use a mean field theory approximation.
Do they force it? Do they force it to be Gaussian?
I believe so.
There's no reason to imagine it would be.
Yeah, right. That's what I'm saying. And so, so the Gaussian approximations can only get you so far. And we use Gaussians because they're tractable, but perhaps there might be other parent families, distributions that are less tractable, but maybe take more compute time.
I've seen people produce bimodal distributions, for example. I mean, that just means there's something wrong with the either the data or the methodology. But it's all right. I'm not keen on these approximations because you're not actually getting the true distributions back.
Of course, you lack data though for too much detail there, so you don't have the information to get a whole lot of resolution distribution, I imagine.
Yeah, I mean, presumably they'll be really broad. But this is what the benchmarking is for, partly to figure out how good is it going to work. So he's using Mike's synthetic network generator, generate large. How big are the networks you're generating?
So far, I've got to 10, works fine.
But how big are they?
Oh, 10 species. Sorry.
Oh, just 10 species.
And then I started doing 20 species. And I think making those models is not hard. It's just that once you start filtering for quote unquote realisticness, then they start dropping like flies. So, and this is also with the whole allostery in it.
Yeah.
So I think that's like the hard part, but
but you got a couple of 20s now, have you?
Yeah, I have like 5 20s.
Oh, that's a bit better. So with the benchmarking, she's going to, she has the ground truth, and then she's going to create experimental data from the ground truth, and then see if she can, with a scrambled network, same carbon backbone, but everything else is scrambled, to see if she can recover the ground truth. The idea being, what happens as you start losing data,
right?
Or some data.
What about robustness to errors?
Exactly. But if there's noise in the data, how does that affect the results as well?
Yeah. One issue that I already see with using the synthetic models is that when I have noise, then it's really hard for me to, because these are measured at steady state, right? But if you have noise, that means that your values are not necessarily at what the model would compute as steady state.
No, that's right.
So that's why, like, the model has to, like, there is a requirement for it for all the values to match up at steady state. And so that's where I'm running into errors with the noisy models. So that's a quirk, I suppose.
Well, this is the problem when you start, everybody has these fancy algorithms. When you start testing them, even on, you should always test them on synthetic systems, I think. Find out what the limitations are. This is the whole thing with flux balance analysis, is that I don't know if there have been any synthetic tests, which is very worrying, I think. But anyway.
Well, why not? I mean, it seems easy to do. It's been around for 50 years almost, so.
I know.
Seems not hard to do it.
No, it shouldn't be. You need to generate pretty large networks, though. And then you have to have your own synthetic growth objective function. But, it could be done, I guess. Should be able to do it. Yeah, okay. That'll be interesting what the benchmarking is.
Yep. Did you have a question, Michael?
You said you got 5 20-node networks. Out of how many?
About, I want to say like 10,000.
Oh,
That's a lot.
you're gonna have to try to improve this. I don't know why we need to figure out why they're bad.
I could probably help you with that.
Yeah,
I don't think
they're bad.
They're bad. It's just I have like these constraints. So like, right, these FCCs, or first of all, they have to reach steady state. They have to reach steady state. If I perturb them 10% or 50%, their FCCs can't, or their concentrations can't be like 1,000. Their FCCs can't be like over 50. And so.
Yeah, but they're just not coming to steady state. I mean, you should be able to figure out what is the problem with the model. And then maybe that can be used to improve the software to generate better models. So, I mean, when it doesn't produce a steady state, some things go to infinity?
I don't know. I just filter them out.
Oh, okay.
Yeah.
Yeah, okay.
In my test, like half of them, maybe half would go to steady state and the others would go to infinity for some reason or another.
You need to do deep dives into some of the models that don't work, Michael.
I wonder if you could do one of those like plots. Like, do you know what a Ramachandran plot is for protein stuff?
No,
I've heard of it.
Okay, basically it just tells you like which angles are possible, which angles are not possible, and there's like a 2D axis. So it would be cool maybe if you had like a plot of like these kind of parameters are not possible, and then but then now that I think about it, like if you have a 20 if you have a 20 species network, then there's probably going to be like 50 parameters, and then that's like 50 parameter or axes. So that doesn't make sense. But
it's constrainable. Can you add constraints to your fitting process? Try and say constrained within these bounds.
I mean, it's not fitting. It's just generating random networks.
Yeah.
So
that's another thing for Michael. What's it called? Package is that you have, or he has minimums. Like, so minimum of like these many sinks or sources or boundary species, but then there's no like constraint above that. So, yeah.
I don't mind going up. But do we have do you have some simple methods for generating, because there's a lot of stuff you have to type to generate one of these synthetic networks, and you have to look at the documentation, and I find it quite tricky to do. Do we have any sort of shortcuts now?
We talked about that, and you put something on Slack, but it included like defining the number of reactions, and that's
Okay. We don't have to
that was
define
an
number.
issue.
Just say roughly 30, or roughly 40. What happens if you just say, you know, a rough number?
You can do a minimum. You can say, well, you can set a minimum for the purely random networks with no distributions.
Yeah, I don't have to be super precise on the number of reactions or species. But it'd be nice to have just a simple method you could call just to generate some basic networks.
Yeah, I can work on that.
Otherwise, it's too hard. I think it's too hard to use otherwise.
Another thing though for me is that, remember how you told me to use those, like, what's it called? The reaction distributions from
Oh,
big
yeah.
models. So I've
yeah.
only been using one template. So maybe I can try other templates and see if those
Oh, I see. Yeah.
templates work better. But yeah.
Okay. Yeah.
One other thing, for your distributions, have you tried something simpler, like just a uniform distribution? If you have bounds, you could just use the uniform. You try something like that.
I don't know.
I've had some
if I'm
success with that
using-
in the past.
I don't even know if I'm using uniform or what I'm using,
Well,
because
you
I
say
just run the same thing
for
again and again.
for your priors.
Oh, the
For
priors.
BMCA.
Yeah.
Let me go back. Oh, I guess I could, but
Which
the
ones though?
reason the reason I don't is because I have, because when you say a uniform prior, you're saying like I don't know anything, right? It's like everything's uniform.
You know the bounds.
But-
That's it. That's it.
Yeah, the trouble is, I don't know if you can do a uniform because a uniform suggests, on average, nothing is likely.
Well, oh, but you want me to have bounds in my distributions for BMCA?
No, I mean, if you have reasonable bounds for the parameters, you could, yeah, you're right. The uniform distribution would then tell you, you know, nothing else but what's reasonable, you know. It's just the simplest approach. And I've had some success with that in a previous project.
Hmm.
Oh, I think
But-
I do the same thing, but I clip stuff.
You'd have to clip them.
I think that's my method of doing this, clipping like the data to like cut off the tails. I'd have to get back to you on that, but I think that's what
I mean, this is where the
maybe you're referring
benchmarking can
to.
be useful as well. You can try different priors on the benchmarking.
Yeah,
Okay.
just thought.
Yeah. All right then. Is that it? Okay, Jen, we're done.
Can I ask a question?
Yes,
Yeah.
of course.
So for the BMCA, is there any other example that have used this method successfully for something interesting?
So I have two examples. Or sorry, there have been two papers published about using BMCA. One is the very first paper, which is kind of, it's benchmarking it, but not with synthetic stuff. It's benchmarking it with like old literature values and seeing like, does this like, do the values kind of make sense? So that I can also put the links in the chat. But then the second paper, they did it on an organism called Yaroia lipolytica, which I don't really know what that is, but basically that microorganism, they put in a pathway inside of it, and then they use BMCA to kind of, I think they looked at the flux control coefficients, but I'm not quite sure. Yeah, let me put the things in the chat. So
Thanks.
mhm.
Okay. Thank you for sharing. Do you have questions? If not, then probably we can move to the next one. Janis, do you want to say anything else or?
I think I'm done.
Okay, I'll stop the recording right now then, and then